So welcome everybody to this H3D Foundation webinar. I'm Nicola Elliott-Wong, the program coordinator at H3D Foundation, and it's my great pleasure to host these sessions. So today we're very blessed to have Paul Jensen with us all the way from California. He's up early and we're very grateful that you're here, Paul. And I'm going to introduce him with his bio. So Paul Jensen is a professor at the Scripps Institution of Oceanography, Center for Marine Biotechnology and Biomedicine, University of California, San Diego. His research interests lie at the interface of marine microbiology and natural products chemistry. His lab studies what marine microbes produce, natural products, where they live, why they make them, and how to better exploit them for useful purposes, including drug discovery. While working with cultured microorganisms is an important aspect of this research, omic approaches are providing important new insight into the extent of microbial diversity in marine habitats, how effectively it has been accessed for natural product discovery, and how environmental metabolomes shape community structure. His recent work includes new approaches to characterize unexplored marine chemical space, while his interests in chemical ecology have helped shape our understanding of the roles of natural products in predation defense. Paul holds a Ph.D. degree from the Scripps Institution of Oceanography, an MS degree from San Diego State University, and a BS degree from the Florida Institute of Technology. Paul, we're so thrilled to have you with us. I know you've been a friend of H3D for some time now, and I know in particular our natural products colleagues are particularly excited about this webinar. I'm going to hand over straight away, but first I'm just going to remind everybody that we're going to have a Q&A at the end, so please save your interesting questions for that time or populate the chat so that we know what to expect. Paul, thank you so much. I hand over to you.
Thanks very much, Nicola, and thanks everyone for joining, and super excited to be able to share some things we've been doing lately with you that I'm really excited about, and so let's just jump right into it. And so I think this group understands the importance of natural products as a resource for drug discovery, so I'm not going to belabor that. Maybe some of you are less familiar with the field of marine natural products, and so this is an area that I've worked in my entire career. And I think you could say that this field really began in the 1950s when people, particularly in Asia, occasionally would get sick from eating certain types of seafood, in particular filter feeding mollusks like mussels, and they wanted to know why they were getting sick. And it turned out that those mussels contain toxins. And so you could argue that was sort of the birth of the field of marine natural products chemistry. In the 70s, people began to realize that you could collect soft-bodied marine invertebrates like sponges and gorgonians, pictured on the right here, and that they were just a wealth of new natural product chemistry. And so a lot of work went into scouring the oceans and collecting different types of marine invertebrates. And to date, more than 40,000 new compounds have been described, really pretty extraordinary amount of chemical diversity. More than 12 of these compounds are approved as drugs. And there's a website here listed that keeps track of all the marine natural products that are in various stages of clinical trials. And so my interests in this field are really focused on microbes in the ocean and asking questions about if marine microbes make new and unusual natural products. And we focused a lot on marine sediments, and we've used various techniques to collect those sediments, including samples from the deep sea. Happy to talk about that if anyone's interested. We focused a lot on a group of bacteria called actinomycetes because, as many of you may know, they're common in soils and have been a rich source of antibiotics. And we were interested in if we could isolate actinomycetes from the ocean and if they might be different from those that have been seen on land before. And some of our early forays into this area, as we started to look at the diversity of actinomycetes we could isolate, we recognized that many of them appeared to be distinct from those that had previously been imported from land. And so some of those lineages are circled in red here. We spent a lot of time focusing on this one group, which we originally called MAR1, which we ultimately described as the first obligate marine actinomycete genus, which we called Selenospora. So that was really exciting for us to show that we could isolate new taxa, new actinomycete taxa from the ocean at the genus level. We studied this genus extensively as a source of new natural products, and this is just a snapshot of all the chemical diversity that we found from just this single genus. I want to acknowledge my colleague, Bill Fennicle, who's an organic chemist at Scripps, who I've collaborated with for many years on natural product discovery. And so really a pretty extraordinary diversity of chemical structures coming from this single genus. The red box in the upper left around Selenosporamide A, this is a very unusual molecule. It's a very potent proteasome inhibitor. And this compound has advanced to phase 3 clinical trials for the treatment of glioblastoma. So we're very excited about that. And just the wealth of chemical diversity that can be discovered by culturing new taxa. So more than half of the compounds we found are new. And I think you can make an argument that if you go to a new environment and you isolate and obtain and culture new microbial taxa, that's one way to access new chemical diversity. I'll also quickly mention another group we spent a lot of time working on. We have called this one MAR4. This one falls within the genus Streptomyces. I'm sure many of you are familiar with this genus, a very rich source of natural products and antibiotics. These were really weird looking Streptomyces. There's a picture of a couple of the colonies here. They tend not to make aerial hyphae. And we were really curious about studying these in greater detail. And so here's the phylogeny of Streptomyces, and here's the MAR4 lineage. So you can see it's really distinct from other Streptomyces, and we've been able to delineate this lineage into 8 different species based on average nucleotide identity. And we found that, surprisingly, this group, this MAR4 group, they're very good at producing what we call hybrid isoprenoid natural products. And so some of you may be familiar with these, and these are biosynthetic hybrid molecules that have a terpene component to them. And I've circled some of those terpene components in red in this slide here. And this is important because, one, bacteria generally don't make these kinds of molecules, so it was really unusual to find a group of bacteria that was so capable of doing that. Prenylation, or the addition of these terpene units, is important in drug discovery. It can increase the biological activity of compounds. And we've done a bunch of genetic analysis on these strains, and we see that the genetic basis for this is that these, on average, these MAR4 Streptomyces contain about 4 prenyltransferase genes per genome, which usually most Streptomyces don't have any of these genes. And so we understand the genetic basis for why they're so good at producing these compounds now. So that was really interesting for us to have another group of unusual marine-derived actinomycetes. They don't only make hybrid isoprenoids. We've also found some unusual polyketides, and here's an example of 2 of them. And so 2 of these MAR4 species shown here are both capable of producing these molecules, which are called indanoperol A and B. And these molecules contain this really unusual pyrroloketoindane functionality, which is shown right here. And you can see the difference between them is this one has 3 chlorines and this one has 2. And so it turns out that indanoperol A is a pretty potent antibiotic. And so interestingly, B has pretty much no activity. It's just missing that 1 chlorine at this position. And so this was exciting for us. It's active against both gram-positive and gram-negative bacteria. We've had some discussion with Susan Winks there at H3D about sending this compound to you for testing, and there was some interest there. And unfortunately, while we're still able to detect the production of this compound, for a reason we don't yet understand, the yields have become very, very low. So we've been struggling isolate a couple of milligrams to send that out for further testing. So we're still exploring that and thinking about maybe using some synthetic biology approaches to try to capture this molecule. But anyway, it's another example of an interesting natural product from one of these MAR4 strains. Another thing we're able to do, which is pretty fun and exciting these days, is by sequencing the genomes, we can try to identify the biosynthetic gene clusters that are responsible for making these molecules. And we do this process that we call paired omics, where we compare the genomic and the metabolomic data. And so if we have a small handful of strains that can make the compound, then we can look to see if they all share a certain gene cluster that would be a candidate for that compound. And in this case, we had a little bit of a heads up because there's a related molecule called indanomycin, and the gene cluster for that was known, and that's shown on top. And so we found a gene cluster that pretty closely matched the indanomycin gene cluster, or BGC, biosynthetic gene cluster, and we were able to bioinformatically ascribe that gene cluster to the production of these indanoperol compounds. And there's some really interesting biochemistry going on here in terms of how this indane ring is formed, and we think there could be a new mechanism for that formation, which is pretty interesting and maybe a story for another day. But we still hope to get enough of this compound to send to H3D for testing, and so something we're still working on. Okay, so everything I've told you about is dependent on culturing, right? We start with a microbe and we culture it and we try to find new natural products. And there's a lot of challenges with this. And of course, we know that most marine bacteria have yet to be obtained in culture. And even the strains that we can culture, when we sequence their genomes, we see that most of the biosynthetic gene clusters they have appear to be silent, so they get lazy when you grow them in the lab, right? They don't have any incentive to make these molecules. And so there's been some interesting approaches to try to get around these obstacles. And in terms of cultivation, people have developed things like the iChip, which is a way to do cultivation in situ. There's been a lot of work adding chemical elicitors to try to initiate the biosynthesis of these molecules, or co-cultivation, challenge one microbe with another microbe to try to trigger them to start producing a compound. And also synthetic biology and genome mining, heterologous expression, these kinds of things. So a lot of great work's been done in all of those areas. But at the end of the day, there are a lot of challenges in natural product discovery by using these more traditional approach. So also, we also know that from metagenomic work, by sequencing environmental DNA, that beyond the things we can culture, there's vast amounts of biosynthetic potential that's yet to be captured. There's been some really high-profile papers talking about that. A couple of them are listed here. A couple of these have been focused on the ocean, just talking about the vast and unexplored biosynthetic potential in the ocean. And even I just saw this paper just came out a couple of days ago, where a group's developed a tool to try to predict the structures of the molecules that you can be detected, or at least the structures of the compounds produced by the gene clusters that can be detected in metagenomic sequence data. So tremendous amount of evidence that we're really missing the ball in terms of capturing the vast amount of natural product diversity that's out there, especially in the ocean. And so this is where I really want to focus today's talk on, and that is the idea that this is really how the process has been done, right? We start with a microbe, and then we look to see if that microbe can make a molecule of interest. And we started to think, well, can we flip the script, and can we start with a molecule and then look to see who's actually producing it? And so we got some inspiration for this from a method called SPAT, or solid phase toxin tracking. And this has been used for quite a while now to monitor for marine toxins, things like domoic acid. And so people who do this monitoring put adsorbent resins out in the environment, and then they elute the resins, and they use targeted mass spectrometry, and they say, they ask questions like, is domoic acid present, yes or no? And should we be concerned about the levels? And so we looked at that approach and we started to think, well, we've been isolating microbes from ocean sediments and finding the natural products that they produce. And should we be able to detect those natural products in the sediments where the microbes live? And that seemed like a fun question to ask. And so we started packaging up some of these adsorbent resins and putting them out in ocean sediments and just asking the question, can we detect microbial compounds? And so the answer to that was pretty exciting for us. So this is a molecular network. If you're not familiar with these, each of these circles basically represents a compound that's detected in a mass spectrometer. If the compounds are related, they have a line or an edge drawn between them, and that's based on the fragmentation pattern in the MS2 data. And so we were pretty excited because, yes, we could see microbial compounds. Here's a lot of compounds in the Nactin series. We know these are made by actinomycetes, so that was great to see. We were really happy that we could see staurosporin. And staurosporin, we know, is produced by one of the species in the genus Selenospora. So we were actually able to go back to those same sediments and isolate staurosporin producing strains from those same sediments. And so that was kind of an informal link for us, but it was pretty fun to be able to detect a natural product directly in nature and then isolate the microbe that actually made that product. But what we saw overall was that most of these nodes or compounds, we didn't know what they were. And so that really got us scratching our head and thinking about if we could take this a little further. And we started to ask the question, can new compounds be isolated and characterized using this approach? And so we teamed up with a colleague at Scripps at UCSD, Ted Malinsky. He's an organic chemist, and he's really specialized on solving the structures of molecules using very small amounts of material, because we expected the yields would be very low using this approach. And so we teamed up with him, and this project was led by a postdoc in my group, Alex Bogdanov, a very talented natural products chemist. And so we decided to scale up the amount of resins that we were putting out in the ocean. So we put more material out and developed a pretty simple way to do that. We can elute those extracts, elute the materials from the resin that were captured and generate a crude extract. In many cases, these extracts were quite complicated and more complicated than we expected. So we developed what we called a microfractionation technique, where we collected fractions from an analytical HPLC using a fraction collector directly into microtiter plates. And that allowed us to do antibiotic assays on the material that we collected, so we could use antibiotic activity to guide the isolation of compounds. And so we found that that actually worked quite well. And then we could make daughter plates, and we could analyze the different wells using mass spectrometry. In this case, using untargeted mass spectrometry, because we want to cast a wide net. And so we were pretty confident we would see a lot of metabolites in one of these molecular networks. What we didn't know was if we would be able to get enough compound to get NMR data and actually solve the structures of those molecules. So that was the big unknown question for us. And so we did a deployment off of San Diego. So here's the city of San Diego off of Point Loma. There's a Cabrillo State Marine Preserve. It's quite a pristine area. We put some of these resins out in this habitat, and Alex was able to, we didn't see any activity in our antimicrobial screens, but he started seeing some masses of interest, in this case, a compound that appeared to have both a bromine and a chlorine in it. So he used the mass spectrometer to guide the isolation of this compound. And he was able to get 50 micrograms, which was a pure compound, which is enough material for us to get good NMR data using a high field instrument and a cryoprobe. And so with the NMR data, he was able to assign the structure of this molecule, which we've called Cabrillo spiral. And so this is a pretty extraordinary structure. It's a polyketide. If it's unusual in many ways, and one, it contains both a bromine and a chlorine on a polyketide skeleton. That's pretty unusual. And boxed in blue here, I have this functional group, which is an alpha beta unsaturated aldehyde. And to the best of our knowledge, this functional group has never been seen before in a natural product scaffold. And so for those of you who work in the field of natural products, you know that finding new carbon skeletons is difficult, and it's really what I would say is the holy grail of natural product discovery. So we were really, really excited to be able to just put some resin out and capture a molecule with this level of structural novelty. And so that really proved the point to us that we can do discovery using this kind of approach. And so Alex went on to go after some other molecules, again, just following the mass spec data. In this case, there was a compound with a M/Z of 328. It didn't have any UV activity, so he used the mass spec data, again, to guide the isolation. In this case, got 40 micrograms of a compound that, again, he was able to get good NMR data, and once again solve the structure of this molecule, which we've called cabriostatin. So cabriostatin is interesting in a number of ways. It's composed of two parts that we know about. The green or right-hand portion is a decanoic acid, and the left-hand portion is, in pink, is the amino acid statine. And statine is actually the pharmacophore of the protease inhibitor pepstatin. And so that got us thinking, well, maybe this molecule is actually a protease inhibitor. And so we teamed up with a colleague at UCSD named Anthony O'Donoghue, who studies proteases, and we asked, in fact, if cabriostatin could inhibit the protease activity of cathepsin. And so here's pepstatin, and so pepstatin is inhibiting cathepsin D, which is a protease, and at all three concentrations tested. And so we were pretty surprised here that our molecule, which is called the new statin here, in a concentration-dependent manner, actually potentiates the activity of the protease cathepsin D. And so we're not really sure what to do with that information. It's interesting that this molecule has this biological activity, and we've also found some other unusual biological activities associated with it. We were able to, through some other colleagues, to test the effects on beating cardiomyocytes, and it increased the amplitude and decreased the frequency of that beating in a way that's analogous to some heart medication. So interestingly, this molecule, while not antibiotic, has some really unusual biological activities associated with it. And so another thing that we can really do with this approach, which is quite new and exciting, is we can start to ask questions about where else these compounds have been observed. And so many of you, I'm sure, are familiar when you have a DNA sequence, you blast it using the NCBI web tool to look to see where else that sequence has been seen. Well, there's now a new tool called MASS, which is essentially kind of like a way to blast your MS2 data to see if a molecule's been seen in any other data sets. And so this is a really valuable addition to the world or field of environmental metabolomics. And quite to our surprise, cabriostatin, we saw it all over the place, and mostly in studies where people were looking at dissolved organic matter, or DOM, in the ocean. And we see that this molecule is consistently seen in these metabolomic studies of dissolved organic matter. And so it clears to be very widespread in the marine environment. We also saw it in a deposit made from a cultured dinoflagellate. And so that got us thinking that the dinoflagellate was probably the biological origin of this molecule. But the people who did this work also deposited a metabolome from the culture media, used to grow the dinoflagellate, and the culture media was made using seawater. And so clearly this compound seems to be in the seawater and isn't necessarily produced by this dinoflagellate. So we still don't know who's making it, but really interesting that this unusual biologically active molecule is so widespread in the marine environment. And so I've told you about two compounds so far that we structurally characterize, cabriastatin and cabriaspiral. There are many, many more. Here's a list of some of them, many of them containing bromines or chlorines. When we look for matches in databases, we find very, very few matches. So most of these compounds appear to be new. Many of them appear to have new carbon skeletons. So we really think this is a tremendous way to access new chemical diversity. And so everything I've told you about so far has been published, and what I really want to focus on now is some new work that we've been doing in this area, which we're really excited about. And so we've made a lot of additional deployments, and most of those have been three areas. One's off the Scripps Pier, where our institution is, Mia's Reef, and also Mission Bay. And so we made a lot of deployments at these three sites. And what we see if we look at this principal component analysis, so here each dot represents a metabolome, and you can see they're colored by location, so you can see the colors are separating. So the different locations seem to have very different metabolomes. And we also see separation by year, squares versus triangles. So Mia's Reef, very different metabolomes. So different times of the year, we see very different metabolomes, even at the same site. So that was interesting for us too. We can put resins out at the same site at different times of the year and capture different molecules. And so we took a lot of samples at the Scripps Pier here. You can see here, we also see some separation by year of deployment. And we made a lot of deployments there. So I want to tell you a little bit about the Scripps Pier. And so this is our research institution right here. And this pier isn't designed for ships to come and dock to, but it's basically a stationary research platform. And there's a lot of instruments that are used to collect data off the pier. There's also a ladder on this side that can be lowered down, and you're able to get into the water and go diving. And so we started deploying these resins off the pier, and we came up with this kind of complicated way to anchor the resins to the sea floor and just have them kind of floating in the seawater where they could capture molecules. And so we did some of these deployments, and Alex once again started seeing interesting evidence of chlorinated molecules in the mass spec data. So he went after this molecule, was able to get 200 micrograms from we put about 100 grams of resin in each of these hoops. And he combined three of those, was able to get good NMR data once again, and once again solve the structure of this new polyketide, which we haven't named, but it's a macrolactam, and in this case it has four chlorines associated with it, as you can see. So this is a hallmark of marine natural products, high levels of halogenation. And so again, another new structure readily captured using this approach. And so back to the Scripps Pier, another thing that this pier is used for is in this building, there's a pumping station, and seawater is taken up by this pumping station, and there's a long raceway, and the water runs down the pier into some sand filters, and then it's pumped up to, we have an aquarium, and it's also pumped into some of the research labs. And so one day some people in the lab say, so this is the raceway where the water runs through, and people in the lab said, well, rather than going diving, why don't we just put some of these resins out in the raceway? It seems a lot easier. And so we said, okay, that sounds like a good idea. Let's give it a try. So we started to do that. And the other nice thing is that this is our labs face the pier. This is the view we have in the morning. So it's not a bad place to have to go to work every day. And we, on occasion, get these red tides. and we can come in the morning, and these things happen really fast. Like one day it'll look like this, and the next day you come in and the water's just completely brown. And so when we see that, we can just run out and quickly deploy these resins in this raceway when the red tide's happening. The other nice thing is that there's a monitoring system that's part of this big coastal observing system. Scripps Pier is one of the locations, and every week there's a group that does a plankton tow and then reports what phytoplankton are present. And here it's just generally said present or absent or rare, but they actually have counts that they do, and we're able to access those counts. And so we realized that this was a nice data set that could really enrich what we were trying to do. So we made a lot of deployments in the off of the pier in this raceway, and you can see these are the LCMS chromatograms, and you can see they look very different depending on when we deployed them. In some cases, these were deployed to correspond to a visible plankton bloom. Again, a PCA analysis. So we didn't have many replicates, but you can see they're colored based on the date of the deployment, and so you can see a lot of separation. So again, the metabolomes are quite different at different deployments, and here's just a heat map showing that. So in this case, dark or brighter yellow indicates higher abundance of specific compounds. And so in cases where we had replicates, you can see they look pretty similar here, here, and that there's generally a set of molecules that's really driving the difference between the deployments that were made at different times. And so just taking a closer look at what we're seeing. And so again, I've been doing this for a long time, and this just blows me away that this is just one deployment, okay? And this is just some of the halogenated molecules that were detected. And so you can see just an extraordinary wealth of what appears to be new chemical diversity captured by just putting a couple of these hoops out for a couple of days. There were only 2 matches in the Dictionary of Natural Products to penaic acid and pentamarine. And one of the things we're really interested in is, can we link these compounds to their producers? And so we're gonna get to that. But first, let's just talk a little bit about penaic acid and pentamarine. And so these are the structures of these 2 compounds here. So again, another very rare class of natural products, this bicyclic ring system. This is the world's collection of compounds that have this system, so it's very unusual. Penaic acid was originally discovered from extracting 10 kilograms of muscle viscera to get 1 milligram of this compound. And again, these were people who were studying marine toxins, and they were able to characterize this molecule by doing that. The pentamarine came from large quantities of a marine sponge, okay? And so again, to get small yields of this compound. And so we found this molecule, which we think is a new molecule in this series. So that makes 5 in total. And so it seems pretty clear we're capturing these in using our resin approach, and they're being found in filter feeding invertebrates. So it's pretty clear that these compounds seem to be of microbial origin. And so the nice thing that we can do is now, we have these time points. We can look at the abundance of the compound and see how that varies over time. And we can compare that to what's being reported in these plankton counts. And so in this case, not all of these peaks where the compounds were abundant were tied to blooms of the phytoplankton. And so we did a more specific correlation analysis, and we're actually able to find one significant correlation between dinophysis, which is a toxin producing dinoflagellate that was significantly correlated with the production of the penaic acid and its derivatives. So we don't have this organism, access to this organism, so we don't know if it's the actual producer, but this gives us a hint at who we should look for to see who might be the actual source of this molecule. We're seeing other really unusual chlorinated sulfolipids, and so here's just the LC trace, and so all of these peaks are linked to different chlorosulfolipids, chlorinated sulfolipids. And so these compounds have never been reported from California before. And so that in itself is a first. And so let's just take a little closer look at two of those. One of them that has 6 chlorines and one of them that has 11 chlorines. And so here's the 6 chlorine compound. And this actually we were able to identify as mitolipin A. And so here's the structure of that compound. 1, 2, 3, 4, 5, 6. Yeah. And so this was originally reported from mussels collected in the Adriatic Sea. So again, from people studying marine toxins. The 11 chlorine compound is shown here again. So another chlorinated sulfolipid. This, we believe, is a record for the most number of chlorine atoms ever reported on a natural product. And so this is a new compound. And the most closely related compound comes from a freshwater chrysophyte, so a type of golden algae. Again, we don't know who's making this molecule, but we're very happy to be able to find it. And the stereocenters, by the way, they're inferred based on some known related compounds in both cases. And so, again, we can look at the relative abundance of these chlorosulfolipids, and we can see there were some real peaks. There was actually a bloom of a diatom called Pseudonychia right around the peak of these compounds. And so that was interesting for us. Right at the same time, there were strandings of sea lions, marine mammals along the coast right off San Diego. It was pretty amazing. They were just coming up on the beach where they shouldn't be and foaming at the mouth and acting really unusual. And so everyone was attributing Pseudonychia is known to produce domoic acid, and domoic acid is known to have these effect on marine mammals. And so everyone was assuming it was domoic acid, but we didn't detect domoic acid at all, but we detected a lot of these chlorosulfolipids. So we're wondering maybe if these are some other marine toxins that might be of concern. And so we contacted a colleague who had grown some a Pseudonychia in the lab and gave us an extract of it, and it did not contain the chlorosulfolipid. So again, really don't know who's making these compounds. We did, again, a more careful correlation analysis, and things really get complicated here. So these chlorosulfolipids come in different chain lengths. And so it looks like the C24 chain length compounds had a strong correlation only with one organism, and that's this diatom, Bacteria stratum. But then the chlorosulfolipids of different chain lengths often correlated with multiple taxa. So pretty confusing story here in terms of teasing apart who's making what in terms of these molecules. And so the last one I want to tell you about is the most exciting one, and this relates to a compound that Alex detected with an MOZ of 381. And so we noticed that the production of this compound, which is shown here, so we can see some real peaks in abundance of the compound, and these were correlated with some major blooms of an organism called Akashiwa. Okay, and this is the dinoflagellate, and these blooms are very intense, greater than 100,000 cells per liter. And at the time that we're seeing such high levels of these compounds. So that was interesting for us. And once again, we were able to do a more sophisticated correlation analysis, and there was only one organism positively correlated with abundance of this compound, and this is this Akashiwa sanguinea. Okay, so that suggested that this might be the source organism of this compound. So what is this compound? Well, first, these blooms are really intense. I mean, this is a picture looking down from the pier into the seawater. So I mean, the ocean really does turn red when we have these big blooms, and Alex was able to use the mass spec data to guide the isolation of this compound, get some really good NMR data. And so this is the first time now we've been able to get, almost milligram quantities of compounds. And so we're pretty excited about the fact that the yields can be so much higher by trying to time our deployments with these blooms. So we were able to get relatively large amounts of material. Again, another new polyketide structure. Here it is shown here. And this molecule has this unusual feature. It's got this gamma-lactam appendage to it. And so this is pretty unusual functional group to have on a polyketide. There are a couple other examples in the marine natural products literature where this type of gamma-lactam has been seen appendage to a natural product, and those have come from different sources like bryozoans or cyanobacteria, or even the sea lamprey pheromone. So again, a very unusual molecule. We don't know anything about its biological activity yet, but we're still interested in asking who is the producer. And so fortunately, we have a colleague at Scripps, Mike Latz, who actually cultured this organism from that very bloom that I just showed you. And he was able to grow up a leader of the culture, and we processed that culture. And so this is what the extract looks like directly from the Scripps peer. Okay, so it's actually two compounds, and they're just stereoisomers. They just differ at this position here, and they're in about equal amounts. And so that's what we see when we capture the molecule directly from nature. Doug was able to separate the cells from the culture broth, and there was just a tiny amount of this isomer in the cells. And then when he looked at the broth, we see both of them. So it looks like they're producing this molecule and releasing it into the media. Slightly different ratios in the supernatant than what we see in nature, and we don't know exactly why that is yet. But anyway, super excited about this because now we've finally really accomplished what we had hoped to do, and that was capture a molecule first and then link it back to the microbial source of that compound. And so really, really excited about the possibilities moving forward of doing this kind of work. So just to wrap all of this up, we know that there's tremendous biosynthetic potential in the world's oceans that's yet to be accessed, and we need to come up with new methods to try to capture that chemistry, and environmental metabolomics is really paving the way to try to come up with these new approaches. And it's not just better instrumentation, it's better access to web tools. Like I mentioned, MASS, there's another one called GMPS. These are web tools that are really allowing this kind of work to be done much more efficiently. We think this direct capture method really has a lot of promise to access new carbon skeletons. And as I showed you, we can readily incorporate bioassays into this whole approach, which can really guide the isolation of molecules for whatever particular interest you have. There are challenges, and resupply is the big challenge. But hey, I'll take a new structure with a really intriguing biological activity, and then be happy to work on that challenge down the road. Linking compounds to producers remains difficult. We've had our first success in that regard, but there's other challenges there as well. And there's also real opportunities to study chemical ecology if that's your interest, using this kind of approach. So yeah, really excited about the future of using this kind of direct capture methods in the field of marine natural products chemistry. So just want to acknowledge the people in the lab, and Alex, as I mentioned, has been doing most of the work. We've got lots of collaborators, and I just want to call out C4D, and I think Hillis van Wezel is going to be giving one of these talks at some point in the future. And he's had a lot of discussions with H3D about teaming up to do antibiotic discovery, and so pretty excited about the possibility of doing that moving forward. And so with that, I'll be happy to entertain any questions that you have.
Thank you so much, Paul, for that very enlightening talk. I'm wondering if there are any questions. Feel free to raise your hand. Godfrey, go for it. I think just double check you can unmute yourself. Go for it.
Yeah, sure thing. Thanks, Paul. Very exciting talk, and thanks for the brilliant work. I was just curious whether you have a plan to move to other coastal sites, or you wish first to exhaust the work around the coast where Scripps is. But this definitely is quite some very impressive work.
Yeah, we have, I feel like we're just getting started in our own backyard, and there's a lot of habitats we haven't deployed these resins yet. What I'm really hoping is that other people will start to use this approach because we'd really like to see it widely applied. And I honestly, I don't think it's only useful in the ocean. I think you could do this in soils as well. You could put them, you could deploy them in soils, and then if it rains, you'd be okay, or you could wet the soil after you deploy them and see what's captured there. So I'd be really interested in hoping that people will also try this approach in environments besides the ocean.
Sure, thanks.
A question from the chat. Many thanks for the excellent talk. I'm not a chemist, but I'm curious about how you were able to measure the masses of two compounds at 40 and 50 respectively. Further, how did these quantities allow you to acquire NMR spectra? My chemistry colleagues usually indicate that milligram quantities are required for NMR analysis. I'm asking because we often struggle to isolate pure compounds in milligram quantities, and it would be very useful for us to understand whether this procedure could be applied in our own work. That's from Dr. P. Moyle.
Yeah, sure. Thanks for that question. So, mass spectrometry is very sensitive. So it's not really a problem to detect these molecules even when they're present in low concentrations by using mass spectrometry. But you're correct. The challenge is getting enough material to do NMR. So you need a high field instrument, and ideally you need a cryoprobe as well. And so in the US at least, most of the major research institutions, have that kind of facility somewhere in the chemistry department. And so, but in the absence of that, then there's some challenges. There are new methods to solving structures using crystallography, like this micro-ED method, where microcrystals are grown. That may be one way to solve a structure with just a small amount of material. But yeah, at the end of the day, you're going to need milligram quantities if you don't have access to a high field NMR instrument.
And over to you, Lone. Go for it.
Good morning, Paul. Hope you're all right, and thanks for a great talk. I was wondering, with all these structures, do you think they all are derived from bacterial, algal, natural organisms in the water or sediment? And how do you differentiate them from molecules that come from anthropogenic sources, human contamination, whatever?
Yeah, hey Lona, great question. And so we do clearly see some anthropogenic compounds. I haven't talked about them because they're not so much of interest for us, but we do see them. Yeah, and I don't know that all of them are made by phytoplankton. Some could be made by bacteria. We only have access to phytoplankton counts, and so that's what we're trying to correlate with. But it would be interesting to try to correlate with other types of potential producers. And, there's the other question, is something I know that you are always interested in, and that is, once these molecules are released into seawater, what happens to them, right?
Yeah.
And are what we're seeing the actual natural product, or in many cases, they could be, these molecules could be biotransformed, if you will. One organism might produce them, another organism might change them in some way. So that adds another level of challenge to try to figuring out who's actually producing the molecule. And so, yeah, I think there's a lot of exciting science that we can do around addressing exactly these kinds of questions that you bring up.
Yeah. Thanks, Paul.
Over to you, Bryce.
Yeah, very nice talk, Paul. And I was very interesting on the side where you use some metabolomic to identify some compound. And I want to understand when you identify some compound with metabolomic, what is the way forward? Do you isolate those compounds, or do you try to couple with the AI tools to first of all identify the promising one that might have a biological activity before isolating?
Yeah, I don't want to be in the business of just isolating molecules for the sake of doing it. We've isolated and characterized a couple of molecules just to establish that we can do it. My interests are much more to try to isolate molecules with specific biological activities of interest. So we really want to focus on using this method and incorporating bioassays to guide us. You know, if you give a natural products chemist an extract that's full of compounds, they're just going to start going after new molecules because that's what they love to do. And we've had some good success with that. But we're moving forward. We're really focusing on antibiotic discovery and trying to use antibiotic screens to find the next great new antibiotic. And as far as AI goes, there are people who are generating some tools now that will help speed up the structural elucidation process. And one of those is called SMART. You still need to get NMR data, but the algorithm with the input is certain 2D NMR experiments, and that can help piece structures together. So I'd like to think that in the relatively near future, there's going to be some AI-assisted tools that are going to help solve the structures. So to take some of the long and time-consuming manual component of that process out of the system.
Thank you.
Could you maybe speak a bit, Paul, to sort of next steps if somebody wanted to adopt these methods or get involved or connect with what you're doing further? What is the sort of access points, either from a personal, I don't know, fellowship perspective or from an institutional collaborative perspective?
Yeah, so a couple of things. So you know, the beauty of this direct capture method is it's embarrassingly simple and it's not expensive. The resin's not hugely expensive. Anyone could go out and capture molecules using this kind of approach. Anyone could start doing some antibiotic screens that are linked to this. So I think it's a very accessible method. In terms of structural elucidation, that's where things get more complicated and maybe collaborators are needed to help that side of it. But certainly this is a way to access chemical space that's previously been inaccessible. And I think, you know, what you access is going to vary dramatically depending on where you are. So there'd be real value in people all over the world trying to capture the metabolomes in their local environments. And so in terms of getting involved in fellowships, you know, we have a program at Scripps. It's called SURF. It's a summer program for undergraduates that students can apply for. And for this summer, it's too late, but for next summer, that opportunity will be open again. And I think, you know, at the undergraduate level, I think there's a fair number of fellowships and things. And then I'm always happy to, if people are interested in applying for various sorts of fellowships to come to Scripps to work on these kinds of things, I'm always open to those opportunities as well.
Fantastic. Thank you so much. Are there any last questions? Please raise your hand. I hope I haven't missed anything in the chat. So guys, I have populated the chat with our links, of course, our networks, GCADA and Rafiki, just so that you're aware of what's going on there. Have a look at the websites. And of course, our webinar feedback form where you can suggest future speakers and also give Paul and me some feedback on how things are going on our side. So then I think it just remains for me to say a big thank you to our participants for joining us today, and a huge thank you to Paul, to also your team in the background for this incredible work, but to Paul for sharing your expertise and passion with us so generously today. We are so grateful. So thank you so much. Any last comments from you, Paul, before we sign off?
No, thanks for the opportunity. It was a lot of fun and looking forward to future connections with H3D and antibiotic discovery.
Absolutely. Okay, thank you so much, everybody, and we'll see you next time. Bye.
Thanks, Paul.
Bye.
Thanks, Nicola. Bye-bye.